Hi, thanks for taking the time to come to my talk today. My name is Rashad, and I'm a member of the Albertson Lab at UMass, where we study craniofacial development in fish. Today, I'm going to be talking about phenotypic coupling and constraint of the craniofacial skeleton across fish taxa. And I know that's a mouthful, so let me begin by explaining a bit about constraint, using this example in great apes. So most great apes have the structure running across the top of the skull called the sagittal crest. It connects to jaw muscles, allowing for a higher bite force, which is important for animals that spend a long time chewing tough vegetation. Humans have done away with this at the expense of our bite force, but it's allowed our brain cases to expand given the lack of muscle and bone around it, and made us more intelligent. And this illustrates a key part of constraint. Trade-offs have to be made. And the degree to which you have to make trade-offs has to do with the dependence of one body part on another. When we expand this to fish, we see this model here where we have the pharyngeal arches labeled 1 through 7 in green. All vertebrate embryos have the pharyngeal arches at a very early stage in development, and they turn into myriad structures, but they stem from the same precursor cells that form along the cranial neural crest very early in development. In fish, the first arch develops into the jaw and associated bones, while the second develops into the hyoid and associated bones, including the serratohyal depicted here. So for the purposes of my experiment, I've picked the mandible and the serratohyal as my two bones that I will use as proxies for structures that develop from these arches. And what we can expect is because they come from the same precursor cells, signals that affect the development of one might affect the development of another. But what we also see is that independence of structures and thus diversity increases over evolutionary time, generally speaking. What we see here is in this pre-vertebrate organism, the pharyngeal gill slits are sort of the same uniform structure without a lot of independence and diversity amongst them. Going forward in evolutionary time, we have now a vertebrate, and in this case, a shark with a rudimentary jaw structure, rudimentary hyoid, and then sort of the same repeating gill arches, but nevertheless more diversity. Going even further, we have the carp skeleton, where we see a much more diverse array of bones. We have the maxilla and premaxilla designed to aid in prey capture. Not depicted here, we have the pharyngeal jaws, which have been diversified for prey processing. Generally, a lot more compartmentalized and specialized bones, the further forward we go in evolutionary time. And so one of the questions I'm trying to answer here is, where do these jumps in independence occur? Over the course of the history of fish, we see large jumps in diversity, and any one of these might have to do with a sudden decoupling event, allowing for that clade to evolve more efficiently while making fewer trade-offs. Because as we can see, fish morphology is incredibly diverse. On the left side there, we have a goby. As you can see, both the serratohyal and the jaw are long and narrow. Both have a high length to width ratio. On the far right, we have a look-down fish where both the serratohyal and the jaw are much beefier with a lower length to width ratio. And then we have a bunch of fish somewhere in the middle, like this stargazer, where the jaw might be narrow and the serratohyal is beefy, as it is in this case, or the other way around. And so to answer some of these questions, I've done a couple things. I've sourced skeletons from the natural history collections at UMass, and taken CT scans off morphosource.com. And in both cases, it's a pretty simple task of isolating the bones of interest, placing them on a blank background with a scale bar, and then orienting them the same way. Then I take these photos of them and upload them to stereomorph in R so I can upload a landmark scheme, which are these points that you see all around them. This is a typical serratohyal on the left and a typical fish mandible on the right. And what these landmarks allow me to do is evaluate every single point relative to every other point, which paints sort of a global picture of shape. And then what we can do is we can plot these in the software called GM Shiny, which works off of Geomorph in R. And what we can see, we can make a couple of predictions about how the shapes of these fish are going to line up. Either the structures of both the serratohyal and the jaw are unconstrained, in which case we see them filling up morphospace relatively uniformly, as we see with the sphere, or they might be constrained to a certain region of morphospace. It could really be anywhere, but the point is it's localized within morphospace. And then when we compare the two, specifically comparing the length to width ratio to see if the shape of one impacts the likelihood that the other is going to be shaped similarly, we can have a couple of relationships, either a strong positive correlation, or maybe a flat line indicating very little relationship between the two, or even maybe a negative relationship where if the serratihyle, for instance, is maybe a bit thicker, then the jaw will be is more likely to be slender. So what do we actually see when we convert this to data? This here is the morphospace grid for the serratihyle. And what you might notice outright is there's all of this blank space in the middle with groups clustered on the right and the left. And it doesn't seem to be particularly, groups can cross over from one side to the other, as we can see, even with small sample sizes. You know, the blue group, in this case, osteophysi, are distributed across the map, but no singular group has any samples in the middle, which is really interesting. None of these groups just visually appear to be super independent from one another. They're generally crossing over each other. But the distribution of our samples is super interesting here. When we move over to the mandible, we see a lot less constraint in terms of how these dots are placed in space. They tend to favor the left side of the grid more. But we do see some separation between them. For instance, Holostae, labeled here in green, which is a more ancestral group of fish, seems to be completely separate from Acanthoropterygii, which is the biggest clade of fish that we have in our sample, and Osteophysi. So that could be an interesting case where maybe they are more restricted in how their jaws are able to develop, which makes sense given how ancestral they are. And for questions about what it means for a dot to be localized in any given part of this morphospace, I'd be happy to answer those questions later. It's a little hard to get into that given the limited time. But we ran a couple of statistical tests here. First, we ran t-tests between all of the groups. And we did find that, as we can see here visually, Holostae was separate from a couple of them statistically. This could be due to limited sample size, but given the samples that we have, they were statistically significantly different. And then when comparing the length to width ratios of the jaw on the serratihyle, we saw the serratihyle tended to be more constrained. As you can see, the box plot here is a bit more squat and condensed. And then plotting the relationship between the two, we saw very little correlation, although there is a slight upward trend here that even without the trend line seems to be pretty evident. So there's a lot more analysis to be done here, and I'd be happy to get way more into it later in the talk, but again, not enough time here. So when it comes to the conclusions and implications, we can ask a couple really new, interesting questions. Why is serratihyle development so constrained? Is it the environment? Is it genetics? When it comes to jaw development, why does it appear that some of the ancestral species are maybe a bit more constrained? Do we need to expand our sample size? So on. This also suggests a contributing factor to fish diversity. The fact that we don't really see a correlation between length and width of the serratihyle and the jaw indicates that maybe they're relatively independent. Fish are incredibly diverse, and maybe this is a reason why. They can adapt to new environments relatively efficiently and while making fewer trade-offs. And if there is a sudden jump in independence, it appears to occur pretty early on, especially when it comes to serratihyle development. Again, with a bigger sample size, we could say this with more certainty. But generally, the only groups that we see being statistically significantly different are the ones that are more ancestral. We had a couple limitations to our study, namely sample size, but then also some really weird bone morphologies. For instance, with this jaw on the left here, it's a more ancestral group, which means that the dentary and the articular process are fused, making our landmark scheme really hard to place. Then we had this jaw here and this serratihyle, which both look very atypical. And then for jaws that were maybe a little bit wider, the orientation that we had them at just didn't really do them justice. So working with a new landmark scheme might be more effective down the road, but some of these are just not compatible with the ones that we had now, and it would have been great to include this diversity. Huge thanks go out to Michelle Gilbert, who's getting her PhD this year in the Albertson lab, and of course, Craig, Dr. Albertson. Thank you both so much for helping me with this whole process. I'd be happy to answer any questions later in the conference. Thank you so much for taking this time.